fecal microbiota, then prebiotic, probiotics, postbiotics, and all, right? So,
Yeah,
Prakash sir is there. Sir, please.
good evening, everyone. Very happy to see you. Most importantly, our close colleague Zayan Bhule, all the way from Hyderabad, is meeting. And this is actually the topic of very important as most of the lifestyle diseases, gut microbiota, you know. So how to define the gut microbiota? Every gut microbiota is the process of altering balance, the diversity of microorganisms or metabolic activity, you know, living in the digestive tract. So in gut microbiota, we are actually looking only microorganism in gastrointestinal tract. We know there are two worldwide projects. Microbiota and human intestinal tract gut microbiota, you know, and through these projects, a lot of
Sir, I think your voice is broken, I think. It's not clear.
yeah, actually internet now is it okay?
Now it is okay.
Yeah, so we are basically looking how we actually can balance gut microflora, you know, using the different strategies like the prebiotics, eating the diet, fiber-rich diet, you know, because this non-digestible fibers like inulin or resistant starch, they actually are helpful. They provide actually the food for the microorganisms that are present in the colon, you know. The bifidobacteria group is there or a bacteroid is there because these are actually the useful microorganisms, you know, which helps to produce the short chain fatty acid. So we actually needs more number of the useful microflora and less number of the harmful bacteria, you know. This is what the eubiotic gut means. What is the dysbiotic gut opposite of that? Where more number of the harmful bacteria are there, the gram-negative pathogens, you know, in such cases. So how we can actually balance this? One, of course, is the prebiotics, because we are actually feeding the microorganism present in our GI tract with the food or feed required for their proliferation. Very easy to understand. Next is, of course, the probiotics. And in a probiotics, we have the major two groups. One is the lactobacillus and another is the bifidobacteria. And you know, the lactobacilli are actually microaerophilic lactic acid producing organisms. Survive in acidic and bile stress, more suitable are colonizes in a small intestine. And they basically actually produces the acidify the GI tract, then digest the food, its absorption, you know, and again suppressing the pathogenic microorganisms. Now, if you see actually the bifidobacteria, which actually we have extensive research on this organism, which actually colonizes in the colon, digests the food which is not digested in the smaller intestine, produces a short chain fatty acid as the energy source. More importantly, now lactobacillus as well as the bifidobacteria also helps to strain our immune system. So this is actually very important component, you know, of these probiotic organisms. And more importantly, the bifidobacteria which we see are anaerobic in nature. And when they actually, you know, they need the pH, neutral pH. So you see in our GI tract, you know, the stomach highly acidic, small intestine acidic. But when we go to colon, you will see the pH is towards neutral and low oxygen, means anaerobic conditions. This is what actually bifidobacteria actually proliferates. And you will see actually bifidobacteria colonizes in a human GI tract in the childhood because most of this bacteria comes from mother's milk, you know, helps to develop or no, build our immune system. Uh, The other two molecules are metabolites, which actually modulates. One is the postbiotic also. Recently, people have actually evaluated this. And we even had one or two roundtable discussion exclusively on postbiotics. Okay. So postbiotic, as we know, that these are bioactive metabolites and the soluble byproducts produced by gut bacteria. You know, and they are actually associated with several health benefits. You know, the metabolites grass status microorganisms in very simple word. Now, how these molecules or organisms actually are useful? So one of this first you will see reason is the barrier integrity. You know, they actually enhances the tight junction protein and hence reduce the gut permeability. You will see the septicemia or bacteremia, especially in the senior citizens or the old people. You know, because of the gut permeability, most of this toxin produced in our GI tract is being infiltrated or transfers in the bloodstream. You know, what is this? My probiotics organisms are actually are good microflora is doing is they actually reduces permeability. They even reduces the inflammation also. Then you will see the metabolic regulations like influence the host weight, glucose control, and this is how actually several of the probiotics are credited with reducing the diabetics, right? Satiety hormones production like PPY is there or GLP-1. You know, very, very, very important reactions. This bacteria actually helps either their metabolites or the whole cells, you know, and all. So I will be very happy to interact on this topic. We will go now directly to Dr. Jayant Bhogle sir also. Let us have his opinion because he directly works on this probiotics and let us know how the probiotic are being used in the gut microbiota modulations or in other words, keeping us healthy. Dr. Jayant, please.
Sir, after you have given such a beautiful and a brief introduction, there's hardly anything I have to say. I wholeheartedly support all the concepts which you have very elaborately explained. My my
Thank
my job is basically to produce the organisms on mass scale. You tell
you.
me, ask me to produce bifidobacterium, I'll produce and give you. Okay.
Wonderful.
Yeah, its repercussions, how it functions, how it works, is for
Hmm, hmm.
you to decide. And once you say something, there's nothing which can go against that. I mean, I
Exactly.
wholly agree with you. The present concept of prebiotics, probiotics, and postbiotics, they all work together, and the immunomodulation and all is brought about.
Yes,
Uh,
sir.
yeah, and it's basically a question of research, which you are very actively doing, and I'm actually involved in producing it in a large scale. So you ask me to produce any lactobacillus, I'll do that. But then if you ask me
Yeah,
to
because
study the postbiotic effects of it, I think you are the best judge of it.
yeah, we have in fact the ongoing project on the
Yeah,
postbiotics.
exactly. That's what we were also talking last time. We have two, three ongoing projects.
Yes,
And I think you were in the US about 3, 4 months back to
yes,
study
sir.
some
Yes,
immunomodulations on that.
yes, sir. Exactly.
So I think you are the best man to say. If you say something, there's nothing against that. I mean,
Thank you, sir. Thank you,
yeah,
sir. Now, another very important we have now, I can tell about this Phenomania cohort project. The
yeah.
CSIR actually has a, a national level cohort project comprising of almost 10,000 employees
Uh-huh.
studying the cardiometabolic disorder. And we are actually looking at the gut microbiota of at least 5,000 people.
Oh,
Uh,
that's a very big thing.
exactly.
Yeah,
And
yeah.
this project, yes, what we did basically is 2 years back, we collected a fecal sample
Mm-hmm.
and did the NGS, and then we have actually the overall microbial profiling.
Yeah.
And through that, actually, we are able to actually identify the happiness index.
Uh-huh.
and which are the dominant microflora,
Yeah.
then their diversity,
Mm-hmm.
richness of the diversity,
Yeah.
and which are the actually among the dominant microorganisms, which are the species actually are very predominant. So our study also showed, now it is actually the data is under analysis stage, manuscript is under preparation. The Privatola copri. Privatola copri has been recently renamed in some other with some other name, but this is what actually the information present is available. So such similar projects on the gut microbiota analysis, you know. So
Yeah.
more the diversity is a
Any
better.
other any other group actively working on that can add something to this discussion. It is
Definitely. So
open. Yeah.
in India, the at the pan India wise, of course, the NCCS group is working.
Yeah.
Dr. Sharmila Mande
Uh-huh.
from TCS, they are working. But of course, the CSR, which is actually looking the gut microbiota, is totally from the different angle. This is more with reference to the cardiometabolic disorder, and that project actually halfway at moment. And in my laboratory, we are actually going to do the probiotic intervention and
Mm-hmm.
see how we can actually increase the richness of these microorganisms.
Sir,
See if the
this is a very big project. I can see a lot of people from
yes, sir,
academics
exactly,
like Dr. Maligi and all. Actually, you can come forward, talk to Dr. Halami and say that we will do a part of it and then give you a feedback. That would be most welcome. Am I right, sir? Because
exactly.
because number
Fully
of
agree with
samples
you.
tested are so huge. You know, if two, three
Yes,
agencies
yes.
or academic students who are doing their dissertation or PhD are involved, it will
Jayanta
add
sir,
to
we can
the
ask
yeah, it will add to the
Jayanta sir, we can
technical.
ask from participant also, those who are working on, in
Exactly.
gut microbiota. Yes,
Exactly. They can take up their
very true.
dissertation. Mali can employ two people there. Two of your students can do dissertation on this. Am I right, sir?
Exactly,
Correct.
yes, yes. Very useful concept. Now,
Yeah.
all this study, of course, one is the basic microbiology, but is
Ah.
the molecular biology, and the
Yeah.
ultimate is the bioinformatics.
And see, since the work involved is very basic, any institute who is into postgraduate who has MSc and all can actually rope in two three students for their dissertation or for this kind of work because the magnanimity of Halami's
Exactly,
project is so big that let us support him technically by
exactly. So
assisting
we have
him.
other
Yeah.
two approaches.
Yeah.
One is the prebiotics from the millet, and we
Yes.
have actually because 2023 was the International Year of Millet, and CFTRI actually has a one millet mission project where we check this millet from dietary fibers from the barnyard millet, and we have actually some preclinical trial in a rat model where we could see the changes in the gut microflora, especially the richness, the diversity among most of these actinobacteria, firmicutes, you know, which is actually very much essential or required for our health. In addition, we have actually conducted one very important experiment that is in vitro fermentation. So we actually take the microflora from the fecal sample, mix
Mm-hmm.
with the millet fiber with that,
Yeah.
and then without the probiotics.
Mm-hmm.
Now, this study actually help us to see how the millet prebiotics are useful to grow in abundance the fecal microflora, use microorganisms. Then with the probiotics, where probiotics actually beside their own growth, they're supposed to support the growth of other useful microflora. So that was another actually the finding. The third one is we look some of this metabolite production like short chain fatty acids
Yeah.
and all, and we could actually see some of this metabolites which are actually associated with healthy aging or healthy living are being produced when we actually give the millet prebiotics plus the bifidobacterial probiotics. Now, this is what the message from this work is that either fecal microbiota transplantation or consumption of millet can restore our gut. You know. So this is how actually is studies are actually are of very much importance these days. That the diet rich in the fiber or fiber rich diet is very much required to have to develop a stable gut microflora, to establish the microorganisms which are required for our better health, and the microorganisms which are actually associated making the count of this microorganism which are associated with the lifestyle diseases, the microorganisms which are actually gives the inflammation, you know. So this some of these important findings we could actually make out from these studies.
There are a lot of academic groups who claim to be working with various aspects of probiotics. I think we should jump to the discussion and then take this to
Yeah,
logical
exactly,
end.
exactly, exactly. That will be more interesting.
Yeah, now we'll throw it open for academicians to come
Sir,
and yeah.
Babu Nair, sir, from I think Lund University, Sweden. I
Uh-huh.
think
Oh,
Prakash Alami, sir, knows. Sir, you know,
yes,
na?
yes, 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 yes.
I
I
was there in
pre-
Anand Agriculture University in JRF in Ahmedabad, I mean, in Gujarat, in Anand Agriculture University, Dairy Microbiology. So
Yeah.
I was
uh-huh.
also there under Dr. J.V. Prajapati.
Oh,
Uh-huh.
yes. I was one of this invited speaker in last year fermented food conference in Kolkata,
Oh,
but
oh.
I presented online, and Dr. Babu Nair was
Subrato,
there.
yes, Subrato Hathi is also there.
Yes, yes. Dr. Sakrota has invited me, but I couldn't go because I had some other assignments same time. Now let us
So,
take
sir,
some questions
one
from the.
very basic question I want to ask. Actually,
Yeah.
for isolating bifidobacterium, which media we are going to use, sir? MRS agar or
Yes, MRS agar with cysteine hydrochloride and anaerobic condition.
okay in anaerobic
Academic
condition.
people should know this better than us.
Or
There are a
otherwise,
lot
there
of
is actually a
there are a lot
selective
of
media also.
claims, people working on probiotics. At least I hear a lot of them.
But maintaining the bifidobacteria, isolating a pure form is not that easy. That also I can tell now only.
Sir, you are there to
Oh.
help them out now.
Yeah, exactly, exactly.
You are there to help them out. If they want to grow in large numbers, I am there to help them out.
Yeah,
But they
yeah,
should come
exactly,
forward with ideas.
exactly, sir.
So we have a different contestant. My participant बहुत सारे हैं यहाँ पे. अनुजा Madhekar is there. Atharva Tilewale. Please introduce yourself. Those who are working on gut microbiota, any PhD scholar or any scientist or any dissertation students in MSc. Anyone is working on gut microbiota, you can discuss with Halami sir. Jayanta sir is also here.
Exactly. So bifidobacteria selective media is the one, but then that you can actually only use for a confirmation.
Hello.
Based
Okay.
on yes, please.
Sir, I have done my dissertation work on the topic of formulation of probiotic syrup, uh, which
Very
is
good.
connected with the gut microbiota, in which
Uh,
I have isolate Bacillus subtilis bacteria for because their efficiency, which have make endospore formation, so they
yes,
will survive in the harsh environment in acidic condition in of stomach. That's why
yes.
I have prepared syrup, probiotic syrup for that microbes. On that
Okay.
cultivation, I have used MRS agar, and
Uh-huh.
uh, in case of prebiotic source, I have used oligosaccharide,
One minute,
fructose.
1 minute. MRS agar for what purpose you have used?
Yes, sir.
MRS.
Why you need MRS for Bacillus subtilis?
Correct.
Yes, sir. I have used Bacillus.
Why? The cost consideration is there, na?
Yes, sir.
That
For
is not correct.
providing growth nutrition, sir.
Hey,
For
whatever
whom?
it is, if it is Bacillus subtilis, you don't need a MRS agar.
Yes, sir. For lactobacillus, we can use MRS. In case of
Yeah.
bacillus, I think I have used a NAM, a nutrient agar media. I have used.
I mean, that's a better way of looking at the things.
Yes, sir. NAM I have used and formulate that syrup. Before that, I have make bile salt tolerance test, in which we can detect that they can tolerate a bile salt
Did you
tolerance.
quote? Did you
Yes,
quote your subtilis
yes,
with?
sir.
How did you quote it on NP seeds?
No, sir. I have not quote.
You're not quoted with
Means
acetate, phthalate, and all.
no, sir. I have not quote.
But then
Simply, I have.
they were acid resistant.
Yes, sir, they are acidic resistance because they can give malachite test, no, which have endospore formation, positive test. That's why I have just prepared, sir.
That's good. That's a good effort.
Okay, sir.
Okay, yeah,
In MSc dissertation, I have
yeah.
done on that.
That's
Yeah, it
good.
is a good
I
approach.
appreciate. Yeah.
But
Okay,
it
sir.
has to be more specific and focused. You
Yes,
know,
sir.
yeah, because Bacillus is not a very commonly used probiotics.
Yes, sir.
Bacillus Lactobacillus. These
Yes,
are,
sir.
but otherwise the common cell organisms are the Lactobacilli
Ye-
and Bifidobacteria.
नहीं
Okay, sir.
डॉक्टर
Okay.
साहब इधर ये रशियन कंट्रीज में there's
Yes,
an
sir.
organism called IP5832. It's a Bacillus
Yes,
subtilis,
Yes,
which
sir.
yes,
is
yes.
used.
Bacillus subtilis is the national or microbes in Kerala states. It will used in environmental and food aspects. That's
Yes,
why I have
yes.
No, no,
taken.
no. See, that
Yeah.
is okay as far as generalized conditions are concerned. But if you are very specific, you should have studied more in detail. Just because
Okay,
Kerala
sir.
government has said Bacillus subtilis, I am using it doesn't serve the purpose.
Yes, sir.
No,
Okay,
you made
sir.
a
I
good
have
attempt.
gained that knowledge in microbiology society
Very
also.
good, very good.
It will be a very informative group. I thought that.
Yes, yes. What you are telling is a very useful information.
No,
Yes,
no.
You know.
sir.
See, there is a story. If you are interested, if people have time, I can tell you.
Yes, sir. Please.
During Second World War, this soldiers used to go after camels in desert and then eat the feces of the camel to stop the diarrhea which they are suffering from. And the organism isolated from that is a Bacillus subtilis deposited with Institute of Pasteur under the name Bacillus subtilis IP5832. So that is the first Bacillus subtilis which is used against diarrhea in the form of camel feces, and now they have isolated it, and there is a formulation based on that. Just for your information, since you are involved in formulating Bacillus subtilis, this is for your information.
So is it quite okay? Your question is answered?
Thank you, sir, for giving such a knowledgeable thing.
Yes, okay.
Informative lecture, is it?
Wonderful.
Any other participant want to discuss?
Hello, sir. May I ask a question?
Yeah, I don't, Anuja ma'am. Yeah.
Sir, I am Anuja Madekar. I am an assistant professor with Department of Microbiology at Ferguson College.
Mhm.
And I've also started with my PhD very recently. I'm working on the isolation of probiotics from soya curd. So
Okay.
my a very preliminary stage of my research work. I had a very basic question. I am currently doing the acid tolerance test for the isolates that I have got, and
Mhm.
following the the protocol which is mentioned in the articles, wherein we use MRS medium, and I adjust the pH to pH 2 and 3. But what I'm observing is when I add HCl to reduce the pH,
Yes,
I see some precipitates forming in the MRS broth. So I am guessing that it is because of the proteins which are getting denatured due to the reduction in pH. So, could anybody suggest me a solution to this, or can I just go ahead with the same MRS broth to continue with my acid tolerance test?
Basically, that is not the method to check acid tolerance. Number one,
Okay.
you don't have
Okay.
don't have to adjust pH of MRS and all. You suspend your organisms in the acidic pH, draw samples at a regular interval, and then plate them out.
Okay. Okay. So acidic.
I think if you go through methods in microbiology, volume 3,
Mm-hmm.
you would get this test. I'm not very sure because I must have done it way back. But then the method of adjusting pH of MRS broth and then checking is not the right way because you'll not be able to quantify that also. So it is
Right.
better you add specific number of organisms to various pH outside, draw the samples at different interval of time and then check it. Am I right, Halamisam?
definitely. Because we are looking for percent survival.
Yeah, exactly.
Percent survival under stress, that's the
Huh?
pH. So you
And
did not
you,
look for the cultivation media pH.
huh?
You will be putting in the buffer actually, or
Yeah,
saline.
yeah.
You know, and then you actually see that at pH 2, 1 hour or 2 hours, because this is how actually this probiotic bacteria has to
Yeah.
tolerate the pH.
And if you are working with classical lactobacillus, very difficult to find, huh? Unless you coat them.
Yeah, exactly. So you will
You
see
coat them
the
with acetate, phthalate, and then see you find a survivability.
acid bile stress is a very actually important
Yeah, yeah.
factor,
I mean,
and most of our
that's the one where you can. One acid test for organisms to survive gastric acidity.
most of our actually probiotics they fail
Yeah, exactly.
commercially. You know,
Anyway, but
so more
you will find the organisms. There's no problem, but you find a right way of testing it
yeah,
and
exactly.
quantifying it. Yeah,
So more important for all the researcher or student, the message is that they use the proper procedure,
yeah,
the well
yeah.
defined authenticated protocol, the protocol which is published in a good journal. Following that protocol and then doing the experiment using properly positive and negative control. These are actually the important. When you are checking for any lactobacillus, better you use lactobacillus rhamnosus GG culture. It is commercially available. Or lactobacillus casei strain shirota. You know, you use that Yakult culture. You know, if you are using the Bifidobacter, better you use Bifidobacter minimum BB12 culture. You know,
Yeah,
because these
but
are
she is isolating from some soya product.
yes, sir. So
So
what I
Yes.
was
Yes.
trying to tell her that she used the standard culture for a comparison of her
yeah,
study.
standard culture for comparison. Yes,
And she used the proper procedure, which is a well-established
that
protocol.
is most important.
Exactly. So same thing, actually, story we will be repeating in our lab also.
Yeah.
When the student perform the experiment. One just to go one step ahead is a simulated gastric conditions.
Uh-huh.
So, so once he has done this test, culture
Yeah,
is performing quite all right, then simulated
exactly.
conditions
Yeah,
can be
yeah.
done. You know. So these two
So
simulated tests, if it is done, then your in vitro experiments are completed.
Acid and bile tolerance are the two acid tests for organisms to survive.
right, sir. So you suggest that I should go ahead with the simulated gastric pH conditions
That is
protocol.
that is
Next,
afterwards.
next
So
one.
That's
first you
afterwards.
check its survivability at different pH at different interval of time and how many percentage survival is there.
Using a buffer adjusted to pH 2
Yeah,
and 3.
exactly.
Okay,
At
okay,
different pH, yeah.
okay, okay.
And you enumerate the organisms before and after.
Right, right. Okay, sir. Thank you very much, sir.
Wonderful, wonderful. Now you are talking from Pune, right? So you go NCCS. Many people are actually
Yes,
working
sir.
on this aspect. I think
There are a
his
lot
name
of people in NCL are also working on that.
in NCR also, no?
Yeah.
So whenever
Nene,
you
if you know Dr. Sanjay, Nene, go and ask him, he'll be able to tell you.
Okay, sir. Okay.
the
Or
more
Dr. Ranade is there.
exactly, or the brand
Or
ambassador of Indian gut microbiota is Yogesh
yeah,
Sauche.
Dr. Gadre is there. You know these
Okay.
people? You know these people?
Dr.
Yes,
Dhotre,
sir. Yes, I've heard their names.
Yeah.
Dr. Dhotre is there in
Sauche
NCCS.
sir is Sauche sir is also
Huh?
Yogesh
Dr.
Sauche sir.
Sauche, another name.
Chowche, sir. Yes, yes.
Dr.
If
Yogesh
you
Sauche
if
is
you if you don't find if you don't find any of them, you can come and talk to me. I'm in Pune for 15 days in a month.
exactly.
Oh, okay, sir. Sir, it'll be great if you could share your contact details or email ID. I can maybe write to you
Yeah,
if
yeah.
it's okay.
Sure.
See, Jain sir is from a company. He is an industry person. So
Okay, okay.
his each and every minute is actually the paid minute. You know. So he is not that easily available. So you have to actually before meeting you have to see. You know.
No, but for such work, there is no problem. So as far as science is concerned, let us share the scientific knowledge we
Wonderful,
have. Yeah.
wonderful, sir. Wonderful. Definitely,
Yes.
it's very great
Yes,
of you.
Yeah.
that's it.
Mm.
Yes, any other participant have a question or anyone want to discuss? Shailendra, Shailya Preeti, Shilpa Raj, Sana Patel, Rushikesh Sankalp,
Hey, don't force
anyone
anybody.
else?
If somebody wants to discuss, it's fine. Otherwise, it's okay. Yeah. Because normally people from academics feel that they are sharing some secret with us.
So there are actually some of these companies which are actually providing the kit or doing this gut microbiome analysis. No, so here is actually the list of such kit, like one MIGQ Wellness 360 test is there,
Yeah.
which actually is quite expensive. They are quoting somewhere at 20,000. But similarly,
20,000 is no 20,000 is no expense.
yes, sir.
For their
MIGQ
PhD,
Discovery
they get so
test,
much stipend or something from the government. 20,000
exact.
is nothing. Yeah.
MIGQ Discovery test is there. This is
Yeah.
actual test for individuals seeking a foundational understanding on the gut health and science backing, starting for improving their digesting wellness. So within a 10,000.
Yeah.
And the
So,
information actually one can get is on the comparative gut health score,
yeah.
then microbiome diversity score. Then you will know what are the most abundant microbes in your GI tract, then their composition, then we will know their metabolic indicators also. Then we will know the personal dietary recommendation based on this information. No, the dietitians or those who work on the gut microbiota, they will be actually able to tell how much dietary fiber we should eat daily. Then personalized probiotic recommendation, whether they have to actually consume regularly the probiotics. Like I take every day the lactovit. Lactovit from the Reddy's laboratory. I hope Jayan sir will agree with me.
Yeah.
Exactly,
Twen-
lactobacillus.
25 years I was inhaling coagulants every day.
Yes, sir.
Okay.
And then you can actually consult the expert based on this gut microbiota information, especially with reference to your the digestive health
Yeah.
and lifestyle decisions. You know, you know. So one can actually easily prevent the lifestyle diseases. One can have a better healthy living, healthy aging also, and as well as they can actually be live longer because finally, you know, The microbiota present in our GI tract, they actually determines how we healthy and how long we are going to live. The longevity of life, in other words. Any other question? If it is there, we can discuss that. Otherwise, we can close also
Yeah,
with the answer
Uh,
recommendation at the end.
that's right.
so I had a question for Dr. Jayant. So
Yeah,
you mentioned about your project regarding the removal of anti-nutrients from soya bean.
yeah,
So, sir, could you elaborate more on it?
yeah. Defatted soybean meal, which is used as a very large protein source for animal feed. You have that idea?
No, no, sir. I haven't come across
Ah.
this
So,
information.
anyway, as a introductory, I can tell you the storyline. Say
Yeah, yeah.
soybean after removing the lecithin and soya oil is left with a soybean cake. It is called as defatted soybean cake, which has a large nutritive value in terms of protein, because soya protein is highest available, and this is given to aquaculture, broiler, all poultry fodder. But it has some anti-nutrients like trypsin inhibitors, then beta-conglycinin, raffinose, and stachyose. So we remove all these anti-nutrients by producing some proteolytic enzymes, then maintaining a acidic pH of the environment so that other microorganisms would not grow, and knock off uh, trypsin inhibitors and beta-conglycinin by more than 90%. Organisms would grow at the expense of raffinose and stachyose, reducing their percentage, and digestive problems are solved. This is a solid state fermentation, where in which all these anti-nutrients are removed by a consortium of organisms, and we have isolated this consortium from the factory where soybean oil is produced.
Okay, sir. Sir, but when you mention that raffinose and stachyose is removed,
Yeah.
but they are the only carbon sources in soya bean, I mean,
Yes,
if
yes.
my knowledge
The
is correct.
organisms will grow at the expense of this carbon source.
Right,
And,
right.
and basically, defatted soybean meal is given mainly as a protein source to fodder than carbon source.
Okay, okay,
It,
okay.
it contains about 48 to 49% protein.
Right,
So
right.
it is mainly given as a protein source. So, and what happens is raffinose and stachyose, if they are present, they interfere with the digestibility of
Right.
chicken or large animals or even aquaculture animals. So once that is removed, the digestibility improves. And there's beta
Okay.
conglacinin and all, they give little allergic reactions. So they have
Hmm.
to be broken down into its constituents.
Also, sir, this soya milk or soya curd, it has
Yeah.
a very peculiar beany flavor. So
That is
is
the character of soya, and I have no knowledge of it. I have no business with it.
okay, okay, okay.
Yeah.
Yeah, but I have the answer for that question, and we have
Yeah,
actually
Dr. Halami will be able to tell you because he's
exactly.
working
Yes,
a
yes,
lot on
doctor.
soya meal. Yeah.
Yes.
So, sir, my
But
question was actually that are these two oligosaccharides responsible for that beany flavor?
yes, yes,
Stachyose and raffinose.
yeah, yes, yes. And then we have actually investigated some of this other metabolite. And then we could hydrolyze those metabolized by the probiotic cultures, and we reduce to 70-80% of this bendy flavor. We have the evidences. And just last
Okay,
week, the
okay.
student was there in the Singapore for a conference. He presented that work. So we are actually pursuing that.
We knock off these two by a culture of consortium of Lechiniformis and settlers.
Yes, sir.
Okay, sir. Okay. Thank you, sir.
Okay, so
Doctor, yeah,
Prakash sir,
please. Yes,
if
yes,
no
please.
more question, then shall we
And
conclude?
let me elaborate very little on that. The
Yes.
the scale of operation is 100 tons a day. Okay. So that is the
Okay.
scale at which the fermentations are carried out in our country also.
Okay.
Okay. So anyone you want to discuss anything? Otherwise, we'll conclude the
Yeah, we
today's
can close.
session.
It's already, yeah,
Yeah.
the time is up.
Yeah. So yes, thank you so much, Prakash sir. Thank you so much, Jayan sir, for a wonderful discussion about the gut microbiota
I
modulation.
I'm uninvited guest to you today. Okay.
No, sir, it's our pleasure that really it was a very good session. And
Yes, sir.
okay, sir, thank you so much. Thank you, Deshmukh sir. Thank you,
Yeah.
Prakash sir, Viprant sir, Upasna ma'am, everyone. Okay, thank you.
Thank
Goodbye,
Thank you.
you.
everyone.
Thank you.
Thank you, everyone. Good evening.